Hi everyone. So as you might have guessed, so yeah, today I will be talking about my work on the ontogenetic variation in coagulotoxicity in this beautiful snake that you see here, the northwestern neotropical rattlesnake, Crotalus culminatus. So quick introduction to this species. So Crotalus culminatus is part of what is known as the Crotalus turisus complex. So the neotropical rattlesnakes, which are found from Mexico to all the way down to Argentina. This species in particular is endemic to central and western Mexico, especially. It is a pretty big rattlesnake. It can grow past 1.5 meters, which makes it one of the largest rattlesnake species in Mexico, and its venom, from what we know, is rich in enzymes known as snake venom metalloproteases, which are generally hemorrhagic or active against the blood clotting cascade. And interestingly, again, like this species has no neurotoxins in its venom, which is strange, considering that neurotoxic phospholipases are perhaps a trademark of the neotropical rattlesnakes as a whole, but culminatus lacks them completely. So the work that I did on that we did on this species back 2 years ago resulted in a paper that was published last year. So you can find it online. Whether I recommend it or not is another matter. But yeah, so let's dive right in and see what exactly we found there. So we analyzed venoms from Crotalus culminatus and also two of its most closely related species, Crotalus mictlantecutli. You know you're a badass when you're literally named after the Aztec god of death, and Crotalus tzabcan from Yucatan, which I have included a photo of here because, my God, those red markings are the stuff of dreams, man. So what we did for the analysis was basically incubating venom with human plasma on this machine that you see in the picture on top, StarMax hemostasis analyzer, which we affectionately call Dracula because it is all about blood. So we incubated the venom with, again, plasma and cofactors, coagulation cofactors, namely calcium and phospholipids, which are necessary for the coagulation cascade to function properly. So we also tested the efficacy of one commonly marketed Mexican antivenom called antivitmin, and inhibitors of metalloproteases, not only SVMPs, but metalloproteases in general, which are called prinomastat and DMPS. And lastly, we also tested for activation of blood clotting factors, which are enzymes that are involved in the blood coagulation cascade by means of fluorometry. So here you see what we found as to the clotting time induced by venoms from several rattlesnakes. And this is a lot of names, a lot of bars. So I just want to draw your attention onto the highlighted ones here, which are orders of, more over an order of magnitude lower, faster than the others. And they all belong to either neonate or juvenile Crotalus culminatus individuals, whereas the adults are much slower. None of them goes under 100 seconds, whereas the neonates consistently range within 10 and 15 seconds clotting time. So why does this happen? So why do neonates clot plasma so much faster than adults? Apparently, it is because they activate one of the main blood clotting factors, which is factor 10, which is directly involved in the formation of thrombin, which in turn cleaves fibrinogen to form fibrin clots. You see here in this graph, which is based on fluorometry data, that a neonate Crotalus culminatus venom activated factor 10 up to 40% the level of an active enzyme control, so factor 10A, which is already active by itself, which greatly outclasses any of the other venoms, including an adult of the same species, which you see right next to it in blue, which didn't even get to 10%. And what we also saw is that this factor 10 activation is highly dependent on the presence of cofactors, of phospholipids, and especially calcium, because you see here, when both cofactors are present, we get the pattern that you can already see on the left graph, whereas without phospholipids and without calcium, the F10 activating action is nearly virtually abolished. And this has repercussions for anti-venom efficacy as well. On the left, here you see graphs of clotting time with venom incubated with antivipmin in blue, and then prinomastat in green and DMPS in pink. Now, you can't see the blue line, can you? Well, that's because it completely overlapped with the venom-only control, meaning that the antivenom didn't do anything to slow down the procoagulant activity of this venom. Prinomastat, on the other hand, did at a concentration of 2 millimolar, especially with a decreasing venom concentration. However, it did bring clotting levels close to spontaneous control, which is usually between 450 and 650 seconds. Whereas for DMPS, you see a lot of variation and big error bars here. Also because we had to increase the concentration of DMPS by tenfold to 20 millimolar for it to have any effect to begin with. So it was not nearly as efficient as prinomastat, sorry. Whereas with an adult Crotalus culminatus, well, this time the curves are much closer. All the curves except DMPS are much closer in space. You see again, antivipmin didn't do much, and prinomastat didn't either, but this time it was probably because the adult venom is not particularly procoagulant on its own, so it doesn't affect the coagulation cascade as heavily as the neonate to begin with. So to recap, we have a clear ontogenetic loss of metalloproteases that activate factor 10, and also a reduced, if not even abolished, procoagulant activity without calcium and without phospholipids, and especially calcium, which has big consequences for assay reliability in a lab setting, because many standard clotting coagulotoxicity tests do not include cofactors in their protocols, meaning that the activities like these might go completely absent in studies that do not include cofactors. Again, antivenom, in this case antivipmin, was not effective in reversing, or at least in neutralizing, the activation of factor 10 by Crotalus culminatus venom, whereas one of the two metalloprotease inhibitors, prinomastat, was. So now that I'm back here and I've started my PhD, I already agreed with my supervisor, Dr. Brian Fry, to continue the work on this species, and especially to try to find out like why do these animals have procoagulant toxins, especially instead of the neurotoxins that are standard for in other members of the Crotalus durissus complex, and why do we see ontogenetic variation? So what drivers lead these animals to lose such a potent procoagulant venom activity as they age? So to try to address these questions, we will analyze a much larger sample of Crotalus culminatus venoms. We have over 20 in stock. It is still to be decided how many we will actually use from across the species distribution, which, as you can see from this map, extends throughout Western and Central Mexico. The dots here are not the sampling localities for our venom, but all the recorded occurrences of the species itself. So we will also look at literature data on diet for this species, which is poorly known, although available information suggests that they are mostly rodent eaters, that they specialize on mammals from a very early age on, so not really any ontogenetic shifts in diet. And also environmental variables. So where do these animals live? Like what elevation ranges, what kind of habitat, what are the main climatic characteristics of their native distribution? All this to see whether to ascertain whether this ontogenetic variation is due to population structure. So maybe it is present in some populations, but not in others. Or again, pre-specializations, maybe there is something, there is still a lot we don't know about the diet of these species, so maybe there is something directly related to diet that drives variation, or even environmental variables, as is known to be the case for other rattlesnake species, such as climate, elevation, humidity, and the likes. Now, from the clinical side of things, we will also upgrade our preliminary tests by testing 3 antivenom products this time, all of them produced in Mexico, and 3 SVMP inhibitors. Antivipmin and prinomastat are still going to be in the mix, but not the only ones anymore, because this data might very well be needed to inform both clinical practice in treatment of for the treatment of bites from this species in hospitals, and production of antivenoms to ensure that available sera actually do neutralize venom even from neonates of this species. And that although bites from neonate Crotalus culminatus are not, at least from the little information that we have, are not particularly likely to occur in nature, for private keepers, for example, this might be more of a concern, because this species is found in the pet trade across the world, even far away from its native range, meaning that even finding antivenom for it might be difficult. And if the antivenom doesn't work, then that defeats the purpose in the first place. So I would like to conclude, I would like to acknowledge and to thank all the people who have contributed to this work, especially my supervisor, Dr. Brian Fry, and Dr. Christina Zdenek, as well as our collaborators at the Instituto de Biotecnología at the Universidad Nacional Autónoma de México in Cuernavaca, and at Instituto Butantan, as well as all my colleagues and friends at the Venom Evolution Lab. So thank you so much for your attention, and I will be happy to answer any questions.
Thank you very much, Lorenzo. Great talk, great topic. And I think I have to invite you then for chapter 2 in a few years, I guess.
I hope so.
So are there any questions from the audience? Yes, Ferenc had another one.
Hello, great talk. I wanted to ask you, because I'm not familiar with exactly, but is the culminatus venom directly used in the preparation of the antivipmin, or just labeled as a possible treatment because of the cross-reactivity?
That is actually a very good question. I mean, the short answer is no, but it gets a little bit more complicated than that, because antivipmin is made, if memory serves, from 3 snake species, one of which is what we used to know as Crotalus simus, so the Central American rattlesnake, which, however, was split into multiple species, one of which being Crotalus culminatus from 2005 onwards. So this also means that since it is often hard to trace back like the exact venom sample used for production of any single vial of antivenoms, we actually don't know which of the species that were originally lumped into Crotalus simus were actually were indeed used for the making of any antivenom batch. And since these species do differ sometimes dramatically in venom activity, that can cause a lot of confusion and yeah, can be another unwelcome variable when it comes to treatment of bites.
Okay, and if I may continue on this path, do you think it would be also wise to include to the antivenom production not only the species, but different life stage of that particular species? So get the venom from the babies and the adults as well, taking into consideration the age-driven intraspecific variation.
I think so. Yes, definitely. And of course, this depends on like what the data say about like how big the danger really is of getting bitten by a neonate Crotalus culminatus, which I suspect is not that great. But again, data about snakebite are always fuzzy, to put it mildly. And Mexico is no exception. So we don't really have like a complete picture of what happens out there. But yeah, at least in principle, at least in theory, yes, I do think it would be wise to include snakes from different age groups in antivenom production, because especially when the venom effects are so rampantly different between life stages.
And sorry, the very last question about this. Do you think this difference is driven by different feeding habit, or what can be the exact reason for that?
Oh well, yeah. This is what we will try to find. At least based on what we know about the diet of Crotalus culminatus, it doesn't seem to be the main factor, or at least not directly, and in the sense that it's not a shift in prey type like it is for other species, such as the Southern Pacific rattlesnakes, or even outside of the Americas for Australian brown snakes, which change from diet based on lizards or other ectotherms to mammals as they age, and so the venom changes because of that. Culminatus appears to be a rodent specialist, or at least a mammal specialist from the very beginning. So that I don't think that is the reason. But again, we don't know yet. It could be. It is possible. I'm not ruling it out. But yeah, we will have to see.
All right. Thank you.
Thank you. So I see there's a question in the chat. Yeah. So yeah, Mike, we can't hear you. Your audio is turned off.
Great. I just talking to myself. It's great. Okay, the next question is from Margareta in the chat. Are you planning to collect pregnant females in all localities in order to collect venom from neonates, or do you have another strategy for sampling this sometimes complicated group? Also, how will you improve collecting data on environmental factors?
Oh, well, thank you, Margareta. So for the samples, actually, we are lucky that we have a great collaboration in place with the Instituto de Biotecnología in Cuernavaca in Mexico, which is even within the range of Crotalus culminatus, and they supply us with venom samples from their own herpetarium and from the wild as I think from the wild as well. So yeah, we have thorough coverage in both geographically and age-wise, thanks to this. Then for also from all the way from neonates to adults. As for the environmental factors, yeah, of course it is. I can see why you're wondering about that, because yeah, we are based in Australia and these snakes live in Mexico, so it's not like we can lace up our boots and just go out in the field and take measurements. That would be a bit impractical, but we can base ourselves on what we know about the distribution of these species and the habitat preferences, which for Crotalus culminatus are range from like montane pine oak forest all the way down to tropical dry forest, and at elevations that span from sea level to 2,000 meters. So we can get climatic data from those areas from databases like WorldClim, for example. But yeah, this is still very much in the works, so it's nothing official yet, and I don't have like a clear I don't have like all the details clear yet, but it's definitely I will, if you like, I will keep you updated on that.
Okay, any other questions? Else I would have some. The PPL you use as a cofactor.
Yes.
Is it an intact phospholipid, or is it more like the products you would get by the activity of phospholipase A's that might be also in the venom?
No. The phospholipids that we use as cofactors, yeah, are phospholipids that we order from our supplier, which is like the same company that provided us with the StarMax hemostasis analyzer. So I'm not really sure like where exactly they come from, like if they are human or other derived from other animals. But yeah, no, they are phospholipids. Not they haven't been touched by venom yet until we incubate them with it, of course.